Thank you very much for a kind introduction, Caroline. Hi, everyone. Welcome to Lipidomics webinar, which is dedicated to skin lipids. It's my pleasure to talk for next 30 to 40 minutes about diversity of lipid species in the skin, their function in skin health and skin disease, and also to present you some research which was supported by data generated by Lipotype. So let's dive into the skin lipids. Where do they come from in the skin? What is their function? What is their diversity? And as you know, skin is one of the largest organs in human body, about 2 square meters of surface, dedicated first and foremost to creating a physical barrier between our internal environment and all the hazards and all the dangers of the outside world. The skin's outermost layer, stratum corneum, consists of tightly packed cells and lipids that prevent the entry of multiple pathogens, allergens, chemicals, and other harmful substances. Skin is also an important chemical barrier. Skin is secreting antimicrobial peptides and other substances that inhibit microbial growth and provide a hostile environment for pathogens. Skin is colonized with microorganisms. It hosts diverse microbiota, which also exerts, in a way, protective action as it competes with potential pathogens and contributes to immune defense. Skin is an important immune organ because it acts both as physical barrier and it's not just a passive shield against pathogens, but it contains a complex network of immune cells which create skin's own immune system. And last but definitely not the least, skin massively contributes to our sensation, to how we perceive the outside world. For instance, skin contains various mechanoreceptors that detect touch, pressure, vibration, but it also is innervated with nerve fibers, which are involved in transmitting sensations that signalize hazard. So you can say whether certain things are hot or cold, whether certain stimuli are causing pain. And it would be impossible to talk about lipids without looking closer at skin's anatomy and physiology. So you can see on this slide a beautifully presented different anatomical components of the skin. So let's look closer at different anatomical structures and talk about the origin of lipids in these structures. On this slide, you see sebaceous or oil glands that are lying in the dermis, a lower layer of the skin, and these glands produce sebum. The function of sebum is to lubricate and make skin and hair waterproof, while also providing antimicrobial protection to protect against pathogens. And this is the first lipid matrix that we encounter on the surface of the skin. If we go deeper into the skin, you see there is an outermost layer of epidermis, stratum corneum, which is consisting of non-viable cells, which are embedded in a very specific lipid mortar. And we are going to be talking about stratum corneum in majority of our case studies today. We are going to talk about its composition and its features in a lot of detail in later slides. In this instance, I would also like to touch a little bit upon the lipid biosynthesis and how it takes place in the skin. So process of lipid biosynthesis begins in keratinocytes with enzymes initiating synthesis of fatty acids from acetyl-CoA. Afterwards, these fatty acids undergo elongation and desaturation process, which creates various long-chain fatty acids, which are especially important for skin barrier function. Afterwards, these fatty acids are used to synthesize sphingolipids, which afterwards undergo a range of enzymatic transformation with formation of ceramides. And ceramides would also be in the spotlight of today's presentation a lot. Concurrently, there is a biosynthesis of cholesterol, which is happening in epidermis, which also massively contributes to lipid matrix and supports barrier integrity. Finally, all of these lipids are organized into lamellar bodies. You see them on the slide, shown as tiny purple granules in keratinocytes. And contents of these bodies is then secreted into extracellular space, forming a very well-structured lipid matrix that has a very specific organization and provides protection against water loss and various external physical, chemical, and biological factors. A few words about keratinocyte differentiation. Keratinocyte differentiation is also tightly interconnected with lipid biosynthesis and complexity of the lipid matrix in stratum corneum. So everything begins in stratum basale, where keratinocyte starts its differentiation pathway. And as it progresses to the top through stratum spinosum, stratum granulosum, stratum lucidum, up to the stratum corneum, keratinocytes lose their organelles. They are essentially non-viable cells at this point. And on this slide, they are presented as bricks that are embedded into the mortar. And mortar is actually a very lipid-rich matrix, primarily consisting of ceramides. And we will talk about this in the next slide. Here, I would like to elucidate the differences between sebum composition and stratum corneum composition, this lipid mortar, which creates a robust barrier between our outside and inside environments. So as you can see on the slide, sebum primarily consists of triacylglycerols, followed by WOX esters, squalene, and non-esterified or free fatty acids, with a small addition of cholesterol and esterified cholesterol and diacylglycerols. Whilst on the graph on the right, you can see what is the composition of stratum corneum. Stratum corneum consists up to 15 to 25% of lipids, and 50% of these lipids are ceramides, followed by cholesterol, non-esterified or free fatty acids, esterified cholesterol, and a very small amount of cholesterol sulfate. On this slide, you can also see the schematic representation of stratum corneum, already familiar brick and mortar model. So I remind you that stratum corneum is a complex matrix which consists primarily of proteins, up to 75–80%, and lipids. And this brick and mortar arrangement consists of dense, anucleate, non-viable, protein-rich keratinocytes, which at this point are called corneocytes, which represent the bricks, and they are embedded in the mortar. There are 15 to 25 layers of corneocytes in stratum corneum, and these 15 to 25 layers can also be sampled and evaluated in clinical studies. So speaking of ceramides, you all are familiar with ceramides as components or ingredients for personal care products. This is a massively hyped marketing area. At the moment, the global ceramide market size is valued to be over $360 million of US dollars. Currently, there's also a very strong consumer interest in purchasing personal care products that contain different types of ceramides. Consumers not only look for products that contain ceramide, they also acknowledge that they are familiar with the benefits of these ingredients, so that more than a half of consumers in different countries, based on the marketing research, are familiar with the benefits of ceramides, labeling them as ingredients that are recommended by doctors or by physicians, skincare specialists, and they also identify these components as biocompatible with the skin. However, with this slide, I would like to sensitize you and to emphasize that ceramides are much more diverse and much more complex than just a part of an INCI list on a cream. So it is known that 25 different classes of ceramides exist as of today. So they are chemically identified, and they have a diverse chemical structure due to the presence of specific sphingosine base and specific fatty acids attached to this sphingosine base. We are able to identify and quantify 12 ceramide subclasses, which are represented on this slide. Why is it important to have a knowledge about the subclass of ceramide? So on this slide, I would like to show you a summarized body of evidence showing the association of different lipid classes to different skin conditions and diseases. And with this, I would also like to emphasize that not only the class of ceramide is important, but it's also important to know the structure of ceramides. As you see in atopic dermatitis, it has been shown that long-chain ceramides are reduced in this skin condition, whilst the amount of short-chain ceramides is increased. And it also has been shown that short-chain ceramides have a little bit different phase behavior, and they're less prone to build these beautiful lamella in stratum corneum, and thus they cannot effectively prevent transepidermal water loss, and they contribute to exacerbation of atopic dermatitis. So how can we actually measure skin lipids in the skin of humans? How do we approach this question, this topic in clinical studies? So the sampling per se is quite simple. It's a very easy and reproducible technique, which is also painless for the subject. There is a preparatory step with large D-squame D100 disc, which allows to remove all of the debris from the surface of the skin. It also enables to remove any potentially present contaminants from personal care products if they were not washed off or the washout period was not sufficiently long. And afterwards, this prepared area is sampled with smaller D101 discs. And for us, it's sufficient to have just one D101 disc in order to extract lipids, to be able to identify them and quantify them. So when we get this disc into our hands, we do perform lipid extraction, and then there is a step of lipid identification with mass spectrometry. And as an output of this endeavor, it is possible to quantify not only ceramides, but also other lipids that are present in epidermis. These are deacylglycerols, triacylglycerols, cholesterol, and esterified cholesterol, as well as 12 subclasses of ceramides, which have been mentioned before. And having said that, extracting and analyzing one small D101 D-squame disc, it's possible to identify and quantify more than 250 individual lipid species in the sample of stratum corneum. So now I would like to move to clinical studies and to practical cases to showcase how mass spectrometry-based lipidomics can support various research questions spanning from basic research to quite sophisticated topics of product development. And we begin with a classical case, which was published in 2017. This is the study which was performed by Lipotype, and it is focused on variability of skin lipids depending on multiple factors. So main research question in this study was, how do skin lipid profiles differ depending on parameters such as sampling depth, sex, age of participants, and is there any variability across different body sites? So for this purpose, there has been a recruitment of 104 panelists consisting of 65 females and 39 males in the age between 20 to 89 years. These were healthy individuals. They did not have any skin conditions or skin concerns. And these were the sampling sites in the study. As you see, the sampling has been performed on the forehead, cheek, shoulder blade, on the chest, on the abdomen, on the volar forearm, on the palm of the hand, on the outside surface of the hand, on the buttock, in the groin, on the thigh, on the central calf, on the foot, and on the heel of the sole. And if we look at the total lipid amounts that resulted from this sampling on diverse body areas, you can see that there were lipid-rich areas and there were areas that were very poor in lipids. So cheek and forehead are perfect examples of lipid-rich areas with very high total amounts of lipids, whilst groin, heel of the sole, palm of the hand, top side of the foot are classic examples of lipid-poor areas. So as you see, the variation is quite high in this situation. There has also been a variation depending on the sampling depth. Remember, there are 15 to 25 layers of corneocytes that are available for sampling with this method. And in this study, the sampling has been conducted up to 20 layers of depth. So you see the study found that the total lipid amount decreased with increasing sampling depth, and highest lipid levels were observed on the surface of the skin, so outermost layers of the skin, which also contain sebum lipids, superficial sebum lipids. And this is visible in this next slide when we also provide more refined information about skin lipid profiles with consideration of different lipid classes. So you see that there is a composition change with sampling depth, showing a decrease in the relative abundance of sebum lipids such as deacylglycerols and triacylglycerols as we progress with the depth of the sampling, and an increase in ceramides in deeper layers of stratum corneum. Speaking of age and sex-dependent variation, among many other things, we found that lipid content of the skin is changing with age in females. As you see on this slide, there is a continuous drop in lipid content in women, while in men, it remains pretty stable and constant at a lower level across all age groups in the study. And finally, I would like to also show you compositional changes on the class level. So you see that there is a drop in ceramide content in women with age, and you see that there is a significant decrease in a total amount of sebum lipids in women with age, and these changes are mainly associated with peri and post-menopause. It's very well described in the literature. However, hormonal status was not evaluated in this particular study. Therefore, we could not establish any correlations here. So this study uncovers that there is a significant age and sex-specific variability of skin lipidome. There is also a variability of lipid amounts and composition depending on the sampling site, and more importantly, on sampling depth. The next study is a very interesting study which addresses the question of skin color and diversity in clinical studies, and it establishes the connection between skin pigmentation and skin ceramide composition. So this is the original publication, and the main question in the study was, are there any differences in skin ceramidome, so skin ceramide composition, in skin with different pigmentation? The study has been conducted in South Africa, and there have been women recruited into the study of different ethnic backgrounds. So there have been albino African women, Black African and Caucasian women recruited, and a portion of participants have undergone sampling on sun-protected area in post-auricular area and on a sun-exposed area on the cheek. So when we looked at lipid profiles of in these participants, it turned out that albino Africans have displayed a higher total ceramide amounts. And when we looked particularly at ceramide profiles, it has been shown that levels of omega-OSL ceramides were significantly upregulated in this study subpopulation. It correlates quite well with other studies that have shown that albino Africans have frequently problems with skin barrier due to impaired corneocyte maturation, and there are also enzymatic peculiarities in ceramide processing enzymes in this population. So this analysis shows that there were wide similarities among different study population in composition and chain length of skin lipids. However, the total amount of ceramides and the amount of omega-OSL ceramides was significantly elevated in albino African population. The next study I would like to present to you explores connection between lipid metabolism and microbiome in the dry skin. This is the original publication, and the main research questions asked in this paper were whether the application of a moisturizer can be beneficial for cosmetically dry skin, and whether this moisturizer can induce functional changes in skin microbiome and lipidome. This was the study design. So subjects with cosmetically dry skin have been allocated to application of moisturizer with free fatty acids and small amount of occluding agents for 5 weeks. And there have been a sampling at the baseline with assessment of skin dryness using corneometry, skin microbiome sampling, and sampling for lipidomics analysis. So these measurements have been done on the baseline and post 5 weeks of intervention. After application of moisturizer for 5 weeks, there have been the following effects observed. First of all, there has been a significant increase in skin hydration and cohesivity of stratum corneum. There was also a significant increase in total free fatty acids and total ceramide amounts after 5 weeks. And these values, as you can see on the plot on the right side of the slide, have positively correlated to skin hydration. There has also been a significant increase in cholesterol amount, especially in the deep layers of stratum corneum, so at the level of sampling between 16th and 20th tape strip. And furthermore, it's important to note that the chain length of free fatty acids has also significantly increased in this study from C14 to C20. Talking about microbiome changes, there has been a significant increase in beneficial S. epidermidis after this treatment, with total bacterial amount not changing. And there have been also some bioinformatic work done on microbial data harvested in this study, and metagenome prediction has demonstrated an increase in microbial production of skin-relevant fatty acids. As you know, S. epidermidis is one of the most important members of skin microbiome, and it plays an important role in maintenance of skin health. Therefore, it's a very interesting interconnection that has been found in there. So in this study, it was possible to show that there is a significant improvement of skin properties and increase in lipid amount and beneficial changes in microbiome in response to application of a moisturizer. The next study I would like to present to you is actually an in vitro study where emollient application has been tested in ex vivo skin model. This is the original publication, and the main research question was, what are the molecular changes in the stratum corneum caused by a cream? It's a marketed product, which was previously proven to significantly improve xerosis, was beneficial in patients with atopic dermatitis and ichthyosis. And the study design, as I mentioned, is an in vitro study where human keratinocytes have been cultivated for 15 days. In between days 9 and 14, there has been an application of a cream to this epidermis, and then a lipidomic analysis in these cells have been performed, as well as spectroscopic evaluation of lipid organization has been performed. So in this study, it was possible to show that the total ceramide amount significantly increased after the application of emollient. There has also been a significant increase in ceramide chain length, as you can see on the right side of the slide. And there has been a significant increase of acyl ceramides after the treatment, which indicates a stronger barrier function. So in this study, it was possible to demonstrate that results from skin models demonstrate improvement in lipid composition and chain length without any alterations in lamellar structure or influence the differentiation of human epidermal stem cells. This was the study design. So primary human cells have been cultivated for 24 hours, and there have been different modulators and inhibitors of differentiation used. The study shows that the main lipids influencing epidermal stem cell differentiations are ceramides and glucosylceramides, so actually precursors of ceramides. These lipids were identified through this lipidomics analysis and siRNA screening of lipid modifying enzymes, and specifically knockdown of two genes. This is ELOVL1 and SLC27A1 led to accumulation of these lipid species, which were found to induce differentiation when added to primary keratinocytes in cell culture. This study highlighted the role of sphingolipids, and particularly ceramides, and hexosylceramides, as well as some glycerophospholipids such as phosphatidylcholines and phosphatidylserines in regulation of keratinocyte differentiation. So this study shows that there have been significant changes in lipid composition during the process of keratinocyte differentiation, and that lipids have signaling function and are controlled by lipid processing enzymes. So to provide a short recap of what we have covered today, so in the first part of the webinar, we have seen that skin lipid composition is very complex and it depends on multiple factors such as age, sex, sampling site, sampling depth. There is a seasonal variation, which we did not cover in today's presentation in the case studies, but there is a significant amount of literature showing that there is a decrease in total amount of lipids in the cold season. This has to be considered if you're planning a clinical study with an intervention. And this is something that has to be taken into consideration. And there is also an influence of skin pigmentation on skin lipid composition. Then we have learned that application of moisturizer can exert tangible positive effects by increasing endogenous synthesis of ceramides, changing the length of ceramides and other lipids in stratum corneum, and by promoting beneficial changes in skin microbiome. And finally, we have covered the topic of lipidomics in vitro settings. And I hope I could convince you that it enables screening of factors influencing skin cell biology, and also lipid changes in vitro could be used for additional product claims if you are in research and development. Now, I would like to say a few words about Lipotype and what do we do. Caroline already mentioned that we are a globally leading lipidomics service provider. The company has started its path in 2014 as a spin-off of Max Planck Institute of Molecular and Cell Biology. At the moment, we are a little bit over 30 people with one-to-one gender balance. And we have also recently opened a subsidiary company, Lipotype Inc. in Boston. Lipotype is GMP certified, so this allows us to work in very strictly regulated environments and help customers to ensure the quality of the data that is produced in the studies. And to recap the topic of product development in cosmetics and personal care, I would like to showcase how Lipotype over the course of the years has supported multiple different R&D projects in this area, spanning from ingredient sourcing, where people are interested to profile their oils or plant extracts when they are choosing optimal ingredient composition for skin with specific concerns. We have supported a number of in vitro studies performed either in cultured cells or in ex vivo explants. And our main focus, our main specialty are, of course, in vivo or clinical studies, where we are mainly working with tape strips, either identifying specific lipid composition in skin with certain skin disease or skin concern, or helping our customers to build claims that are related to either marketed products or products that are about to be launched to the market. With that, I would like to thank you for your attention, and I'm more than happy to take any questions.